Welcome, everyone, to this eSymposia Career Roundtable event. I'm Heather Gerhardt, Coordinator of Diversity Initiatives at Keystone Symposia, and it's my pleasure to moderate this event today. Supporting the next generation of research leaders is a key aspect of Keystone Symposia's mission and history, and we're really pleased to be able to continue this tradition in the virtual format here today. Before we get started, I'll go through some instructions on how we'll be running this session. First, please keep your camera and microphones off during this first portion of the event. We'll start with a 30-minute discussion with our panel, and then we'll open it up to discussion for audience questions in the second half. You can feel free to submit your questions at any time into the Zoom chat. During the audience Q&A, I'll select questions from the chat and ask the person who submitted the question to turn on their video and mic and ask the speaker your questions live. After asking your question, please turn off your microphone once again. And we do ask that you please leave the Zoom chat clear for submitting questions. Okay, and now we're ready to welcome our panelists, and they span diverse career backgrounds and experience. I'd like to invite all of you to turn your cameras on, all of our panelists. Great, wonderful to see your faces. All right, so I'm going to start with having each of you introduce yourselves and tell us a bit about your career trajectories. I wonder, Dr. Maquatt, if you'd be willing to start us off.
Hi, I'm Lynn McQuatt, and I am a professor of biochemistry and biophysics at the University of Rochester Medical Center in upstate New York. I am also founding director of our Center for RNA Biology. So I'm a mechanistic RNA biologist. I study human disease. And also, I am founding chair of Graduate Women in Science to address the leaky pipeline that typifies women, where, as a rule, fewer women who earn a PhD degree go on and use that degree in a career compared to men. So in terms of my career path, I can tell you that I was an undergraduate at the University of Connecticut in Storrs. I grew up in Connecticut, and I started working in a lab between the summer between my sophomore and junior years, and just kind of got very enthralled with the aspect of doing experimental research. So I went to graduate school at the University of Wisconsin in Madison and earned my PhD in biochemistry. There I worked on the E. coli lactose operon, so I worked on bacteria. And then when I did my postdoctoral work at Maquat Laboratory for Cancer Research, I started working on human diseases. And I felt that my very strong background in biochemistry, even though I studied E. coli and did molecular biology, not genetics with E. coli, that served me well in transitioning to studying human diseases. And I started with the beta thalassemias and bone marrow aspirates, and showed human disease could be due to a splicing defect. We're talking a long time ago, 1979, probably before most of you were born. And then worked on the beta zero thalassemias, which were the first examples of diseases due to nonsense mediated mRNA decay. And that's what I'm most known for, working on the mechanism and understanding what NMD does to protect ourselves from the possibility of generating a truncated protein that could be toxic. And also cells use NMD, nonsense mediated mRNA decay, as a way to regulate our genome under changing conditions during development or differentiation, for example. I started my lab in the Department of Human Genetics at the oldest comprehensive cancer center in the United States. It's called Roswell Park Cancer Institute. And after 19 years there, I moved to the University of Rochester, where I am today. I liked being in a human genetics department. but I missed the full range that academics can give one. And so at Rochester, it's a private university, we have the medical center and the School of Medicine and Dentistry. That's where my lab is. But across the street, we have the College of Arts, Sciences, and Engineering. And I missed being with humanities colleagues. We have the Eastman School of Music. I'm very musical, so we interact with those colleagues as well. And that's what I like about Rochester.
Wonderful. Thank you, Dr. McQuatt. Dr. Chang, could I have you introduce yourself and say a bit about your career trajectory?
Sure. So thank you, and great to see everybody virtually. So my name is Howard Chang. I'm a professor of dermatology and genetics at Stanford University. So my background is a little bit different. So I was an undergraduate at Harvard College, and I think I was thinking about potentially a career in medicine. But in my sort of summer between freshman and sophomore year, I had a chance to work in a lab, and that really got me hooked on laboratory-based research. And so I subsequently joined the MD PhD program at Harvard MIT, and I did my PhD research at MIT working on in biology, and I was working on mechanisms of programmed cell death. So it's a very different topic area. Then I completed my clinical training in dermatology at Stanford, and then had an opportunity to kind of join really during the sort of early days of functional genomics, working with Pat Brown and David Bostine for my postdoctoral research. And so I'd be happy to talk about kind of the balance between, let's say, for folks involved in this kind of MD PhD, between clinical practice versus research. But one of the reasons that attracted me to dermatology was because I felt that it was an area where I can really see the biology in action. And that appealed to me, and that the tissue was very accessible. It's very amenable to doing research. And so in my postdoctoral research, I started using these new genomic technologies, looking at programs, gene expression programs that skin cells and different kinds of cells could carry out. And after my postdoc, I joined the faculty at Stanford. Then we basically transitioned from initially looking at tools that would look at known mRNA coding genes to more unbiased methods, tiling arrays initially in those early days. That would basically help us look at the entire scope of transcription, let's say, or regulation from a chromosome or even the entire genome. And so my lab then, we were involved in kind of some of the early findings, discoveries of long non-coding RNAs, their pervasive transcription, and the studies of their function. And my lab now currently is involved in just really trying to understand information content in the non-coding genome, both through non-coding RNAs and other kinds of mechanisms. And along the way, I've been fortunate to be involved in also co-founding several biotech companies. So we'd be happy to chat about that as well.
Wonderful, thank you. Dr. Marcia.
Yeah, thank you, Ether. Yeah, I'm Marco Marcia. I'm a group leader at EMBL, currently located in Grenoble, France. So it's great to see you all from so far away. A little bit sad not to meet you all in person. But yeah, that's where I'm talking to you from. And I reached Grenoble following a trajectory that brought me a little bit across a large portion of the world. I'm originally Italian, was born not very far from where I am now, but I then moved to study still in Italy at Bologna University, where I had a course of pharmaceutical chemistry and technology. And that's during my undergrad studies that I got fascinated by small molecule, macromolecule interactions, and of course of fundamental importance for pharmaceutical chemistry. And I decided to study this more closely at more higher resolution. And so I undertook an internship during my undergrad years at the Paul Scherrer Institute in Switzerland. It's an institute not very far from Zurich, which is famous because it hosts one of the most powerful European synchrotron. This is a large facility infrastructure for many different type of research in many fields, but for what concerns life sciences and biology, particularly to perform X-ray crystallography studies. And I joined there for 2 consecutive summers the lab of Fritz Winkler, who were expert in studying the atomic structures of membrane proteins. It was a challenging time for the characterization of membrane proteins still when I joined their lab. And so that's where I entered the field of crystallography, high resolution macromolecular studies, and in particular membrane proteins. And this inspired me to join then the lab of Professor Michel in Frankfurt at the Max Planck Institute for Biophysics to undertake my PhD in biochemistry with a joint degree at Frankfurt University. And I joined Hartmut Michel's lab because having connected to the world of membrane proteins, well, he was the reference person at that time for this sort of study. He had pioneered the field, obtained the first high-resolution structure of a membrane proteins in the late 70s, early 80s. And therefore, I was eager to learn this field in his lab. And at the time, I worked on a bacterial membrane protein involved in bacterial respiration and also conserved with homologs also in higher organisms like human, where it is involved instead in detoxification, because it's a peculiar enzyme that metabolizes and takes energy out of sulfide ions. It's particularly abundant in bacteria that thrive in volcanic areas. And so that's the time when I, of course, got acquainted with lab work and particularly with structural biology approaches. And I didn't want to lose this expertise during my postdoc, but I wanted to expand my expertise at the same time. And so after completing my PhD, I decided then I had a chance to join the lab of Anna Pyle at Yale, still in the field of structural biology, but this time applied to RNA research. And I worked for a few years on the characterization of a large ribozyme that is involved in splicing the group 2 intron. And so I had this dual expertise on protein and RNA biochemistry and biophysics. And when I had the chance to set up my own independent lab here in Grenoble about 7 years ago, I decided to combine these 2 expertise and yet move again a little bit to a new field, gene expression regulation through epigenetic mechanisms, and inspired largely by the work of Howard and his discoveries of link RNAs and their mechanistic regulations. I decided to apply my structural biology expertise in the characterization of some of these targets, and that's what my group is currently working on, both the RNAs themselves and the protein partners that are regulated by these transcripts.
Wonderful. Thank you. Thank you, Dr. Marcia. So these were great introductions. I think it really frames moving forward with some panel questions. As a reminder to our audience, feel free to enter your questions to the chat and we'll address them later in the session. But for now, we'll turn to our panel. One of the questions I like to start with is, what's the most important piece of advice that you'd impart to our trainees today in terms of choosing a career path and in particular becoming successful on that path? Would either of you like to jump in?
I'd be happy to start with that. And I think a really important advice that I got early on was about kind of choosing the question that you want to tackle. And that's certainly an important issue in sort of academic science, or what scientific question do you want to pursue? But certainly in industry, the question would be like, what's the topic area that the company is going to work on? What product area? And this is actually a little bit different from what people typically kind of think about your training in graduate school, because the kind of things that you learn about is you read a paper and you kind of think about what are the right controls, what are the right, are the data sufficient to support the conclusions and so forth. But when you're choosing a question, the problem is we don't know what kind of answers we're going to get. And we're basically making bets on what areas are really going to pay off, right? And so obviously choosing the right question has a huge impact on whether basically this line of research will be successful, whether it's the answer that people will care about, whether the technology you choose is the right approach to answer that particular question. And I think the framework that was useful for me to think about was that people talked about certain kinds of risk, like for example, that, okay, we think about just a new topic, and I just mentioned that we don't really know, maybe there are a number of assumptions, and maybe we would carry out this research and we wouldn't get to the answer. Okay, so there's that kind of risk. But there's actually a different kind of risk, and that is that you'll carry out the research, you'll get an answer, and then nobody would actually care that much about the answer, right? That's a different kind of risk that I think many students, maybe trainees, tend to undercount, right? So they kind of think about like, well, is this going to work? Are all these things known? Are all these tools in hand? Well, basically, I think when a question is like that, kind of clear cut and obvious, then oftentimes you get to the answer and it's like, wow, people kind of expect that already, or basically there are 3 other groups with the exact same finding, and so it's just basically it's just like a mad rush, right? It's not really fun. And so I think that choosing the question that's completely new, which may seem very scary because we really have no idea where we're going to end up. In fact, you actually mitigate that second risk because it's like, well, it's really new, so whatever you find is interesting. And so that's something that I often think about when I try to talk to students about, well, but this is a potential area of investigation, also for new faculty members to potentially think about.
Well, so most of you are already in graduate school, but what I think, to add to what Howard is saying, I think it's important to choose your mentors wisely. And one way to choose your PhD mentor, which you probably already have done, is to look and see where the graduate students, or if you're looking for a postdoc, where the postdocs go next. You want to have a mentor, unless it's someone who's very young and starting their own lab, and that has its pluses too, because that person is going to be at the bench working side by side with you. You want to look at track record of a trainer. And if you're unsure of an academic career, if that's right for you, you can always test the waters. So when it's time to look for a postdoc, or when it's time to look for a faculty position, you can look at both options. You'll be able to do more in industry with a postdoc on top of your PhD than with a PhD alone. And you'll be able to see what each venue will offer you. So you can always test the waters, see what the fit is like, trust your gut. Different industries have very different cultures, and small biotech is going to have a very different culture compared to J&J, a big biotech company. I would be sure you talk to people one-on-one in those environments when you're checking them out or possibly interviewing. Doesn't hurt to interview to get the best understanding of whether this is a good fit for you or not.
Yeah, I actually feel Howard and Lynne are giving excellent advice. I feel embarrassed of being in an awkward position, being both a mentor here as a panel member, but also the mentee of my colleagues that are more senior. From my more junior perspective, perhaps I can report on one specific experience I've had with one of my very junior lab member who came for a traineeship in my lab a couple of years ago. And to whom I think I gave a good advice, and that is of encouraging this person to not be afraid of a bit of serendipity in the choices that one makes in one's own career. This person finished undergrad studies, and then was hesitating with a number of internships in different labs, and that's how she contacted me, and I could meet her. But because of being insecure of what would be the best lab to join, or the best path, whether industry or academia, at some point one needs to take a next step, especially when one has the right competencies, the good training, the excellence and good knowledge of being flexible and potentially suited to many alternative career paths, and needs to take the risk of engaging in making one choice, moving forward, and see where the serendipity of life takes them. And because it's in this way, I think that doesn't mean that one needs to take unconsiderate choices, but that's the way by which one goes out of their own comfort zone and explores perhaps very interesting paths. And as Lynn said, there's always the chance to then redirect one's choices, but it's important to move on and not hang just because one is afraid of what is the best answer for himself.
Great. It's interesting to hear both a deliberate, intentional path is important in terms of choosing the right question, choosing your mentor, but then also this making space for serendipity. Great advice. Thank you. So how about the biggest challenges you may have faced in your career so far? Maybe talk a little bit about how you got past it, what worked, what might not have worked in addressing this challenge. Maybe you're still in the midst of them. I don't know. Someone like to run with that one? No challenges?
Okay, I can say something. So when we were aiming to understand the mechanism of nonsense-mediated mRNA decay, and this is in my own lab, we had findings that some people had trouble believing. So this was a long time ago, but we knew, for example, that intron position within a pre-mRNA determined which premature termination codons in the processed mRNA were triggering mRNA decay. And, splicing, we proposed, must deposit a mark on newly synthesized mRNAs that persists until the first run of translation, because translation is how you recognize premature termination codons. But introns can affect, we proposed, the processed product made by depositing a complex that persists. And the connections between the nucleus and the cytoplasm were also of concern to a number of people. And I remember hoping, I was young, hoping that we were right, but it was very important for us to do several things to maintain as much credibility as we possibly could. One was, don't overinterpret the data. So I think you can shoot yourself in the foot if you're very dogmatic about a finding. And you propose, remember, you're proposing models. These are models built on the results you've obtained in the context of experiments. So don't overinterpret your data and ask questions about your model from more than one perspective. And be sure you know what you know, and be sure you know what you don't know. I think that's very important. And if you're doing any kind of science at all at one level or another, you're going to experience the same thing, where you have a result and you think the simplest interpretation is X, but you're not 100% sure, or maybe it's not with the current dogma. So you need to tread carefully, but securely with what you know and what you don't know. And we were right, by the way, but it was a little, I was always worrying. I'm like, oh my God, I hope we're right. Because I would go to meetings and I had to ask who this person was after when you're young, can I have a 12-minute talk? So I asked for a 12-minute talk, and a person stood up after I gave my talk and stood up. That's impossible. So I had to ask who that was, and I won't say who it was, but I have to say it was difficult, but perseverance is key.
Thanks for sharing that
Yeah, I can also chime in with some other kind of another example of a kind of a challenging situation that one may encounter. I think a pretty challenging time was when I first started my lab, right? You might imagine that you transition from being kind of primarily at the bench to now kind of like motivating and recruiting people. And I remember that the first group meeting I ever had in my lab was that the minus 80 minus 20 fridge door was stuck and we could not open it. Okay, so the entire meeting was based on dealing with how do we open and close this minus 20 door. And I think John Wren was actually my first postdoc. He might remember that meeting. So I was like, wow, okay, well, so from there, right, the only thing we can do is go up, right, in terms of the quality of things we could potentially do. But even when that was going on, then you have, at the same time, you have to juggle your time. You have to write grants. You have to try to get external support. So all these things are happening at the same time, right? So I couldn't be there opening, closing fridge doors for people. So in terms of this, and then at that time, like the process of writing grants is actually, I think, it's a learned skill. Okay, so I think for folks interested in academia, something that it's good to kind of have, if your faculty mentors will let you kind of review their grants or help you write, that's even better. But my particular situation was that I had worked in the labs of very senior scientists, and the style they write grants is not the same as what a junior person can do, right? And so I've seen some of these grants. I was like, okay, this is what I'm going to write. I'm going to say I'm going to do these things, and that's it. And basically, basically my initial grants were like, they were people would give you very harsh comments, right? And what helped was that then I had another chance to write a grant together with another faculty member. And he basically gave me line by line corrections, like, you can't say this, that's an assertion. You can't, you have to back up everything you want to do. And that was a very useful experience to kind of learn, okay, this is what you actually kind of have to do to kind of convince people. And so I think this would be I think for any trainees who are thinking about writing for fellowship proposals or writing grants, you should hopefully get some other person to read it and then give it to them like a month in advance, right? Because if they're really gonna help you, they're gonna give you like line by line changes, then you're gonna need time to actually execute, to implement those changes.
Yeah, we have a system set up at Rochester where we read other people's grants who are just starting out, and it's easier to write a beginning grant than it is to write your first competitive renewal. And so that's where we find faculty members need the most help. But it's mentoring, as Howard said, at this level is key.
Yeah, I agree with all suggestions from my side. Perhaps not specifically a challenge, but feature of my career that is presenting pros and cons, as you have gathered probably from my introduction. I've moved quite dramatically throughout different fields of biochemistry, starting from looking at membrane proteins involved in bioenergetics and respiration, cellular respiration, then splicing ribozymes, and now epigenetic complexes. And so at times I find this interdisciplinarity in my profile very fascinating, because it has helped me keep my mind open and made me acquainted with various fields. At times, though, this is presenting some challenges, because, of course, especially early on, moving across many communities, many scientific communities, means that you're not necessarily, every time you need to re-establish your interaction networks and to refine your people of reference to give you the right advice, to establish collaborations. So this is a balance that one needs to carefully consider when planning his or her own career path.